Hello. In this video, we're going to be looking at the life cycle of SARS CoV-2. First, let me remind you that you want to start by looking at your detailed learning objectives, so you know what we need to know for the exam on SARS CoV-2. So let's start by looking at coronaviruses in general. Now, coronaviruses are enveloped viruses, and they have what's called a positive sense single-stranded RNA genome. We'll see what that means in a few minutes, but basically a positive single-stranded RNA genome is one that can be directly transcribed into viral proteins. That is, it functions both as a genome and as a messenger RNA. That's what the positive refers to. And that's surrounded by a helical nucleocapsid. The coronaviruses belong to a viral family called the coronaviridae. And there are 6 species of human coronaviruses that typically, and 4 of those 6 typically cause mild symptoms like the common cold in children and adults. And in fact, about 15% of common colds are caused by these 4 species of coronaviruses. The 2 remaining species, however, can be much more severe and cause even fatal infections. They include MERS-CoV-2, SARS-CoV-2, and then there are 2 strains of SARS-CoV-2 called SARS-CoV and SARS-CoV-2. So let's look at the 3 potentially harmful ones here. SARS-CoV stands for Severe Acute Respiratory Syndrome coronaviruses, and this was responsible for a pandemic that occurred in 2003, but it hasn't really circulated since about 2004. Then we have MERS-CoV, and that stands for Middle East Respiratory Syndrome coronaviruses because it was first reported in Saudi Arabia in 2012. But it had rather limited human-to-human transmission. And then SARS-CoV-2, Severe Acute Respiratory Syndrome coronavirus 2, was the one first reported in Wuhan, China in December of 2019. And this virus is characterized by a very rapid and virulent human-to-human spread. And of course, that's the one responsible now for the pandemic we call COVID-19, which stands for coronavirus disease, first reported in 2019. So we're going to be concentrating on the life cycle of SARS-CoV-2, the one responsible for COVID-19. Let's take a little bit of a look at the structure of this virus. Now, as we mentioned, this is an enveloped virus, and it has what's called a positive sense single-stranded RNA genome. Now, you may remember earlier in this unit, we talked about positive and negative strands of nucleic acid, which simply mean complementary strands. When a positive strand is copied, it makes a complementary strand called a negative strand. Now, a positive or sense single-stranded RNA genome is called a sense strand because it not only acts as a genome, but it also functions as messenger RNA, and that can be directly translated by ribosomes into viral proteins. So here's an electron micrograph of SARS-CoV-2. It has an envelope. The nucleocapsid is inside, and it has these rather large spikes, the S protein, that extend from it, and it looks kind of like the corona surrounding a sun, so these were called coronaviruses. And this is an illustration on the right of the same virus. So in gray here, we see the viral envelope, and there are 3 distinct structural proteins that are found in the envelope: the E protein, the S or spike protein, and the M protein. So let's take a look at these different proteins. Now, the S or spike protein is the one that form the corona, the glycoproteins that stick out from the envelope. These are the spikes on the surface, as we said, and they're composed, the spike protein can be cleaved by a host cell protease into 2 subunits called S1 and S2, and both are quite important. It's the S1 subunit of the S protein that actually binds to host cell receptor called ACE2 enzymes on the surface of many human cells during viral adsorption. So that's the protein subunit of the spike protein that allows the virus to adsorb to susceptible cells. The S2 subunit is a fusion peptide, and that allows the viral envelope to fuse with host cell membranes during viral entry or penetration. We'll talk more about that when we get into viral adsorption and penetration shortly. The M protein in the envelope stands for membrane protein. And this appears to be a central organizer for coronavirus assembly. This helps determine the shape of the viral envelope. It binds to other viral structural proteins. It helps to stabilize the nucleocapsid N proteins, which we'll mention down below, and helps promote viral assembly or maturation. So it plays a number of important roles in the assembly and maturation of the virus. The E or envelope protein, which we see here, also plays a role in the production, maturation, and release of the virus. And this is especially common where maturation of the virus is going to occur, again, as we'll see when we get to viral maturation shortly. So those are three structural proteins associated with the envelope of the virus: the spike protein, the membrane protein, and the envelope protein. Now the N or nucleocapsid protein forms the viral capsid, and during assembly, these N proteins bind to the RNA genome of the virus, and they play a number of important roles in the process. They help in viral replication, and they help determine host response to the viruses. Now, these are all structural proteins because they're part of the viral structure: the S, the M, the E, and the N. But in addition, SARS-CoV-2 produces 16 non-structural proteins, or NSPs for non-structural protein. These are functional proteins that play important roles in the life cycle, but they're not part of the viral structure. And 15 of these non-structural proteins form the viral replication and transcription complex, or RTC. And this is the complex of enzymes that's responsible for viral RNA synthesis within the host cell, proofreading the viral RNA, modifying the viral RNA. Some of these non-structural proteins, or NSPs, also play important roles in helping the virus to evade host immune responses, and also in modulating host membrane into viral replication organelles, which we'll talk about in a little bit. So those are the various proteins that the virus needs to replicate and to form structures of the virus. So now let's take a look at the life cycle of SARS-CoV-2. As we learned when we talked about the productive life cycle of animal viruses, the first step in any virus life cycle is the virus adsorbing or binding to receptors on the host cell. If the virus can't adsorb to a host cell, it can't get in and it can't replicate. And this is the role, of course, of the S protein we mentioned, or spike protein sticking out of the envelope. That's what's going to enable the virus to bind to human ACE2 enzyme that's highly expressed on many cells in the body. Now, this ACE2 enzyme plays an important role in regulating blood flow and in blood pressure and such. So it has an important role in many cells in the body, and it's expressed on many cells. This ACE2 enzyme is highly expressed in cells of our respiratory tract. That includes cells of the upper respiratory tract, where the virus usually gains a foothold, type 2 alveolar cells in the lungs, but this ACE2 enzyme is also found on enterocytes, cells found in the ileum and colon. It's found on cells in the kidneys, in the bladder, and myocardial cells in the heart. So because this ACE2 enzyme is on so many different human cells, people that are infected with SARS CoV-2 not only experience respiratory problems like pneumonia, which can lead to acute respiratory distress syndrome, or ARDS, that we talked about back in Unit 3, but it can also lead to disorders of the digestive tract, the kidneys, and the heart. And some of these can be fatal or lead to long-term damage. But first, that S protein has to be cleaved by a host cell protease to become active. It's divided into two subunits, S1 and S2. And as we mentioned previously, the S1 subunit is what allows the virus to absorb to the ACE2 enzyme, the receptor on human cells for absorption. And then the S2 subunit has a fusion peptide, and that's what's going to allow the viral envelope to fuse with host cell membranes. So let's first take a look at the role of the S protein in viral adsorption, which we see in Figure 2. And as we see here, the S protein sticking out of the surface of the virus will bind to ACE2 enzymes on the surface of many types of host cells, again, including cells of the respiratory tract, the gastrointestinal tract, the genitourinary tract, and the heart. And here we see an electron micrograph of a cell infected with SARS CoV-2. This is the cell, and these are the virus particles adsorbing to the cell. Now, once the virus absorbs, that is, the next step is penetration, where part of the virus will enter the cell. And SARS CoV-2 appears to enter two ways. Now, we learned back in the productive life cycle that enveloped viruses can enter two ways: by fusion of the viral envelope with the host cell plasma membrane, where part of the virus enters, or by endocytosis, where the whole virus is engulfed and placed in an endosome. So let's take a look at the two ways of the virus entering. So number one, entering by fusion of the viral envelope of the host cell plasma membrane. Once the virus absorbs to ACE2 enzyme with its S1 subunit of the spike protein, the S2 subunit is a fusion peptide that allows the viral envelope to fuse with the host cell plasma membrane. And that allows the viral nucleocapsid to enter the cytoplasm. And we see that below in Figure 4, Step 1b. So here we see the virus is absorbed. The fusion peptide S2 allows the viral envelope here to fuse with the host cell membrane, dumping in the viral nucleocapsid, the plus single-stranded RNA genome covered with N or nucleocapsid proteins. And then the second way the virus enters is through endocytosis. So in this case, once the spike protein absorbs to the ACE2 on the human cell, the whole cell is engulfed, and that pinches off and places the whole virus in an endosome. And now the viral envelope fuses with the membrane of the endosome within the cytoplasm, dumping the nucleocapsid into the cytoplasm. So there we see viral penetration by envelope fusion, dumping in the nucleocapsid, or the entire virus being engulfed and placed in an endosome, and then the viral envelope fuses with the endosomal membrane, dumping the nucleocapsid into the cytoplasm. At that point, the virus is ready to replicate. Now, again, the virus has a plus single-stranded RNA genome and a plus sense strand. The sense strand refers to the fact that the sequence of nucleotides contain the codons that directly code for viral proteins. So a plus sense strand is one where our viral messenger RNA can be directly translated by ribosomes into viral proteins. Now, to replicate RNA, requires an enzyme called RNA-dependent RNA polymerase. And as we learned earlier on in the unit, an RNA-dependent enzyme is one that copies RNA and produces RNA polymerase. So it's going to copy an RNA strand into another RNA strand. So what the RNA-dependent RNA polymerase does is it copies the sense strand into a complementary antisense strand. Now, the antisense strand is a complementary copy of the plus sense strand. In other words, if there's a thymine on the sense strand, there would be a thymine on the antisense strand. And so the antisense strand cannot be transcribed into viral proteins. Only the sense strand can. But to replicate the RNA, it has to make an antisense strand, and then the RNA polymerase can then copy the antisense strand back into more sense RNA genomes. And it's also going to allow this virus to produce some other RNAs that we'll talk about a little bit later on that help to code for various parts of the virus and these non-structural proteins. But here's the general idea of how a single-stranded plus RNA genome replicates in general. So again, the sense strand here, the positive sense strand, can function both as the genome and can function as messenger RNA up here, which can be translated into proteins. But the virus has to make many copies of the genome. So what it has to do is use an RNA-dependent RNA polymerase to copy the sense strand into a complementary negative antisense strand. Then the RNA-dependent RNA polymerase can copy the negative antisense strand into more copies of the sense RNA genome. So that's how it makes many copies of the genome. It has to make a complementary antisense strand and then copy that back into the genome. And that's also functions then as the messenger RNA, which can be translated into viral proteins. But there's some more complex things that occur here. You don't have to know a lot of detail about this, but I do want to go through a little bit of this to show how SARS CoV-2 does what it has to do. So there's a little description of it here, but keep in mind there's two things the virus has to be doing here. It has to make more copies of its genome. So this top strand here is the positive sense RNA genome. And it has to make many copies of that, so all the viruses that are produced contain this genome. But also what it's going to do is make a lot of subgenomic messenger RNAs. And these are smaller messenger RNAs that code for the different structural proteins, like the S protein, the M protein, the E protein, and the N proteins, as well as the non-structural proteins, or NSPs. So eventually it has to make all these smaller RNAs that code for different parts of the virus and different non-structural proteins needed for viral replication. So one thing the virus is doing is making more copies of its genome because, again, each of the viruses has to contain a genome. So the way it does this, again, is that an RNA-dependent RNA polymerase binds to the 3' end of RNA. That's the end that has the terminal hydroxy or OH group sticking out. The other end, the 5' end, is one that has the phosphate sticking out, as we learned when we talked about DNA and RNA earlier in the semester. So to make the genome, to make copies of the genome, the RNA polymerase binds here at the 3' end, copies the entire genome into a complementary negative antisense strand. Now, RNA-dependent RNA polymerase can now bind to the 3' end of the negative antisense strand, copy that back into genomic RNA. So once the genome is copied into a negative antisense strand, RNA-dependent RNA polymerase keeps copying that over and over again into more genomes until there's enough genomes for all the viruses to have one. But also the virus has to make a number of structural proteins and non-structural proteins. The structural proteins, again, become part of the virus, like the S, M, E, and N proteins, as well as the non-structural proteins that play roles in the virus being able to replicate. And the way it can do this is through kind of a strange process called discontinuous transcription. And what that means is that this RNA genome has a lot of transcription regulatory sequences, or TRSs, located along the genome here. So these are all TRSs, with one at the very end, near the end of the molecule. Now, what RNA polymerase can do is it can bind to the 3' end, and it can stop copying at any one of these TRSs along the way, and then jump down to the final TRS and copy the end of it. So let's say it starts and binds here, the RNA-dependent RNA polymerase. It copies it to the first TRS, then it jumps from here down to here and winds up making this complementary strand. So again, it's just copying a little bit of this, making a complementary antisense strand. Or the RNA-dependent RNA polymerase could start here, but then go all the way down to the second TRS, and then jump down here and copy the rest, and that would make this antisense strand. So it's copied from here to here, and then tagged on the end part. It could copy from here and go to the third one, and then jump down to the end and copy the rest. And that would lead to this antisense genome. And it can stop at any one of these and then jump to the end. So as a result, it makes all of these subgenomic negative antisense strands. And then when RNA polymerase binds to these antisense strands, it makes the messenger RNA coding here for the structural proteins and for the different non-structural proteins. So by having a single genome like this and using this discontinuous transcription, it's able to make multiple small subgenomic RNAs that can be translated by ribosomes into the various structural proteins and the various non-structural proteins needed for viral replication. Now, something else that's been found fairly recently is that early in SARS CoV-2 replication, there's an interaction between some of these non-structural proteins and host cell factors to form some unique structures inside our cells called double membrane replication organelles. And this is where the endoplasmic reticulum forms these organelles surrounded by 2 layers of membranes. There are various types of these. Some of these are called double membrane vesicles. Some are called convoluted membranes. But these form naturally inside of cells during the SARS CoV life cycle. So what some of these non-structural proteins do is they interact with host cell proteins and cause the endoplasmic reticulum in our cells to make these double membraned organelles where we think viral RNA replication is occurring. For example, these are the double membrane vesicles here. These are convoluted membranes. And the viral replication and transcription complex, the sequence of enzymes that are needed to replicate the viral RNA and proofread it and modify it, are found associated with these. And they found replicating double-stranded RNA inside of these various double membraned organelles. So although we're not sure where all of the RNA synthesis occurs in the infected cells, it has been shown that double-stranded RNA molecules, which are thought to be an intermediate in the replication of coronaviruses, segregate into these double membrane organelles. And they have been shown that RNA synthesis is occurring within these double membrane organelles. And it's thought that the advantage of this to the virus, by having this double-stranded RNA, which is functioning as a replication intermediate, we think, in the virus, is then protected within these double membraned replication organelles. Now, you may remember when we talked about body defense against viruses, stimulating innate immunity, that one of the common PAMPs found in viruses are viral RNA viruses is double-stranded RNAs. These act as PAMPs, or pathogen-associated molecular patterns, and they bind to pattern recognition receptors in our cytoplasm to initiate innate immunity. So one of the first ways we recognize that a virus, an RNA virus, has entered the body is that most RNA viruses somewhere along the way produce some double-stranded RNA, which is not normally seen in human cells. And so these act as PAMPs, and these bind to pattern recognition receptors in the cytoplasm to begin innate immunity and lead to the production of interferons and things like that. And so the thought is that by sequestering this double-stranded RNA of the virus inside these double membrane organelles, the pattern recognition receptors out here in the cytoplasm never see the double-stranded RNA. They don't see the PAMP, and so innate immunity is not activated, but the virus can continue to replicate its genome and produce the subgenomic RNAs within these vesicles. So by blocking the initial innate immunity, that means we can't remove the virus as well, which gives the virus a better chance to replicate and continue. Then we get to our next step in the viral life cycle, viral maturation. Remember, that's where the parts of a virus assemble around the genome, and finally release of the virus from the infected host cell. So the small subgenomic messenger RNAs that code for the nucleocapsid protein are translated by cytoplasmic membranes, cytoplasmic ribosomes, where they bind to the genomic plus single-stranded RNA genomes to form the viral nucleocapsid. And we see that in steps 1 and 2 of figure 8. So in step 1, some of the small subgenomic RNAs are the messenger RNAs coding for nucleocapsid proteins. These attached to cytoplasmic ribosomes are translated into nucleocapsid proteins, which then attach to the genome of the virus. Now, also other small subgenomic messenger RNA, step 3, coding for viral structural proteins like the S, M, and E proteins, as well as the non-structural proteins, are thought to be translated by ribosomes in the rough endoplasmic reticulum into proteins that enter the ER. So these are the small subgenomic RNAs that code for the M protein, the S protein, the E protein, the non-structural proteins. Many of these enter the endoplasmic reticulum where they become embedded. They're translated by ribosomes associated with rough endoplasmic reticulum, where they enter and become embedded inside the endoplasmic reticulum. Now, what happens next is that the endoplasmic reticulum, step 4, packages these proteins and transports them to the Golgi by way of an intermediate called the ER to Golgi intermediate compartments, or ERGIC. So some of these pinch off, they form this endoplasmic reticulum to Golgi intermediate complex. And what that does is take in the nucleocapsid of the virus, pinches off, and then that becomes part of the Golgi. And then in step 5, the Golgi will then package the virus, which now has obtained its envelope from the ERGIC, into smooth secretion vesicles. So once that pinches off, this becomes the intact virus. The Golgi then packages the virus in the smooth secretion vesicle, and the smooth secretion vesicle fuses with the host cell plasma membrane, and the virus is released by exocytosis. So this is a virus that's released by exocytosis. And here we see an electron micrograph of SARS CoV-2 being released by exocytosis. Now, one of the hallmarks of severe COVID-19 infection is the development of cytokine storms. And back in Unit 3, when we talked about bacterial pathogenicity, we saw that an excessive inflammatory response can occur when there's overproduction of pro-inflammatory cytokines. And in severe cases, this excessive inflammatory response can in fact be fatal. And we see the same thing is happening with COVID-19. So of course, one of the hallmarks of severe COVID-19 infection that requires often people to go on respirators and can be fatal is when it leads to acute respiratory distress syndrome, which we talked about back in Unit 3 when we talked about an excessive inflammatory response. This is where you have acute inflammation in the lungs, where there's damage to capillaries in the alveoli, and there's pulmonary edema. As plasma flows into the lungs, the alveoli become fluid-filled, and that prevents oxygen exchange. So as the virus is replicating in alveolar epithelial cells in the lungs, those cells become damaged, and that is thought to stimulate a strong, dysregulated pro-inflammatory response, where you get this very high degree of inflammatory response, but the regulation system that tries to dampen that doesn't work properly. So there's a release of inflammatory cytokines that promote vasodilation and inflammation. As the cells are damaged, they create DAMPs, or danger-associated molecular patterns. And so this occurs again when injured or infected cells produce factors called DAMPs that, like PAMPs, can trigger the production of inflammatory cytokines and chemokines. So now this inflammation and the production of chemokines attracts alveolar macrophages. They produce more inflammatory cytokines like TNF-alpha, interleukin-1, interleukin-6. It also produces chemokines, which, remember, attract defense cells to the area, things like interleukin-8. So an excessive production of inflammatory cytokines is where we have the cytokine storm. So some of the inflammatory cytokines like TNF-alpha and interleukin-1 increase capillary permeability and expression of adhesion molecules on leukocytes and on the inner wall of capillaries, so diapedesis occurs. And remember, diapedesis is where defense cells like phagocytes leave the blood vessels and enter the tissue to go to the infection site, and chemokines then attract those defense cells into the alveoli. So the increased capillary permeability in the alveoli enables plasma to flow out of the capillaries into the alveoli so they become fluid-filled. That we call pulmonary edema, or an accumulation of fluid in the lungs. And of course, as the alveoli become fluid-filled, that blocks gas exchange. Oxygen is not absorbed into the blood, and of course, that's going to lead to lack of perfusion. So that's what results in the labored breathing, or dyspnea, shortness of breath, hypoxemia, or low levels of oxygen in the blood. And keep in mind that at high cytokine levels, neutrophils and macrophages can dump their killing chemicals, killing more alveolar cells. Cytotoxic T lymphocytes are also killing the infected cells in the alveoli. That leads to further damage to alveolar cells and lung cells, leading to more release of DAMPs, more release of cytokines, and the cytokine storm just keeps amplifying and continuing. It also results in less production of surfactant, and that can lead to the alveoli collapsing. And the damaged alveolar cells are also releasing vasoactive agents like leukotrienes that lead to constriction of smooth muscles and can trigger bronchoconstriction, production of prostaglandins, which increase capillary permeability, induce fever, also constrict smooth muscles, cause pain. And that leads to more inflammation, more infiltration of fluids, more attraction of defense cells, and this then eventually leads to tachypnea, or abnormally rapid breathing. When you have hypoxemia or low oxygen levels in the blood, then the lungs breathe more rapidly to try to bring in more oxygen, and it also leads to tachycardia or an abnormally rapid heartbeat. And collectively, all of this is what leads to ARDS, or acute respiratory distress syndrome. So again, it's a matter of the virus triggering the production of inflammatory cytokines and chemokines, infected cells being killed, releasing DAMPs, which then trigger more inflammation and bring in more defense cells. Chemokines attract the phagocytes to the area, and when you have this dysregulated production of cytokines, we call that a cytokine storm. And eventually, because of lack of oxygenation of the blood, we get the labored breathing, we get the pain, we get the rapid heart rate, the rapid breathing rate, the lack of oxygenation of the blood, and eventually ARDS. Now, at the same time, it's been found that some of these non-structural proteins of SARS CoV-2 appear to dampen the production of type 1 interferons. And remember, it's the type 1 interferons that are the key part of our innate immunity to initially limit viral replication. Remember, these interferons inhibit viral replication within the infected cells, and they protect uninfected cells from becoming infected. They also help to stimulate the production of the cytotoxic T lymphocytes and the NK cells that we use to kill infected cells. So this initial production of type 1 interferon is key to preventing viral replication and getting rid of the infected cells. If the viruses dampen the production of type 1 interferon with these non-structural proteins, then that's going to decrease our innate immunity, and we're not going to be able to remove the virus as well, which gives a chance of the virus to replicate much faster and survive and cause more severe disease. And then these double membrane replication organelles that we mentioned, produced by the endoplasmic reticulum that enclose the viral double-stranded RNA, are thought to create a protective microenvironment where the virus can replicate its genome and produce the subgenomic RNAs needed to make viral parts. Because the double-stranded RNA is a major PAMP for us recognizing that a virus has invaded the cell. But this double-stranded RNA is recognized by pattern recognition receptors in the cytoplasm of the host cell. And so by sequestering the viral double-stranded RNA in these protected double-membraned vesicles, we don't initiate an early innate immunity, and again, we don't fight the virus as well. And so we have the life cycle of SARS-CoV-2, and we also have, of course, our concept map that helps you to review that material.